is doing a brief overview of meiosis from kind of like a high level perspective. What do we start with, and then where do we end up with? And then we'll do a few mono-hybrid crosses together and see where we're at with that. But I'll definitely need feedback from you all, either in the chat or verbally. You can use your little hand raise and lower. That is helpful as well. And it looks like Julius is joining us as well. Hey, Julius.
Good morning, ma'am.
Morning. We are just getting started. And I'm going to do a very brief kind of high-level overview of meiosis, and then we're going to do a mono-hybrid cross together. So generally speaking, with meiosis, I like to use a human chromosome number of 46. And what that means is if we took one of your skin cells today and did a genetic analysis of it, unless there was some sort of genetic issue, we would expect to see 46 total chromosomes. And then sometimes we refer to those as having 23 pairs. So 46 total or 23 pairs. And that just means that if we were talking about chromosome 21, for example, everyone would have 2 copies of that. You get 1 copy from your mom, and you get a second copy from your dad, and your body reads those 2 copies together to produce a protein, for example. And so when we're talking about meiosis occurring, we're starting with a cell that has 46 chromosomes. And at the end of meiosis 1, we're going to have 2 cells that have 23 chromosomes. Something to keep in mind is that these chromosomes are of what I call the 2X type or 2X variety. And what that means is that in meiosis, DNA replication occurs. And so that happens during interphase. So this is actually a 1, meiosis 1. But during that process, DNA duplication occurs. And what that really means is, let me grab a marker. Your chromosomes are made of 1 piece. During DNA replication, you get a second piece right behind it that's identical, and it's stuck together. So what ends up happening is your chromosomes go from looking like, excuse my drawing, something like this, that's 1 piece, to something like this that's 2 pieces, and it's stuck together here in the middle. So that's really a terrible drawing, but one way to think about it is, do I have one piece and my chromosome is just 1 singular piece, 1 singular marker, or is my chromosome made of 2 pieces and I've got 2 identical pieces stuck together? And so what then happens in meiosis II is that each of our cells divide again, and this is only happening in sex cells, so egg cells and sperm cells. And so we get another round of division. So this is meiosis II. But this time DNA duplication does not happen. So a lot of times meiosis is said to have only one division, I'm sorry, only one DNA duplication, but 2 rounds of division. And so what's happened at this time is we've separated those pieces. So in meiosis II, I've got my 2 pieces and I'm separating them into different cells. And so we now have 4 cells that have 23 chromosomes each, but they're of the 1X type now. So I've just got one piece again. There's a lot of genetic variation in each of those cells that gives us the differences that we see in brothers and in sisters, for example. So this is what we're starting with in meiosis. The DNA is duplicated, so we go from a 1X chromosome or a 1-piece chromosome to a 2-piece chromosome, but we've split our cell. And then in our second round of division, we separate the X again, and we're just back to our 1 piece. Any questions so far?
Yes. Can you repeat what you said about the 2 rounds of division? I missed part of that.
Yes. So my fabulously crooked arrows here indicate that we've got meiosis I that you can think of as the first round of division. The other set of arrows here are meiosis II, which is the second round of division. So meiosis has 2 different rounds of division, whereas if we think of mitosis, that only had 1 round of division.
Okay.
And we end up with a lower number of chromosomes because we duplicated the chromosomes once, meaning we went to this 2X type,
Right.
but we did not do that again in meiosis II. So we have that that content or that second piece that was halved. So one DNA duplication, but 2 divisions, which gives us the 23. In terms of kind of making sense of this, if we think of kind of the result as egg and sperm cells, if we do 23 plus 23, what does that give us?
46.
Exactly. So we're back to the 46, and that's what happens during fertilization. So that would be our fertilized embryo that the baby then grows from. So this definitely connects to kind of the circle of life type of things. Julius, Pooja, do you have any other questions so far?
No.
Sorry, was that a no, Julius?
Yes, ma'am.
Okay.
Questions right now.
Okay. All right. So that's kind of the big picture of meiosis. And what that leads us to, remember we had the 23 plus 23 equals 46. And so our genetics problems, the start of them, are looking at, well, if we take this certain characteristic, maybe we're looking at coat color in guinea pigs, can we predict what the ratios or percentages of offspring based on certain characteristics of the parents? So a lot of this is Punnett square work. Oops. And so with setting it up, so we could say, oops, that's not what I wanted. So if we know in guinea pigs that brown colored coat is dominant, we can designate that with an uppercase letter. Maybe the alternative is tan. So maybe the lowercase is tan. Ah. And so what we're really saying is the phenotype that we're looking at is tan or brown. So the phenotype is the outward characteristic. A lot of times it's visible, not always, but it's what we're coding for. The genotype is the letter combination that we're seeing here. So the genotype are the letters, the phenotype is going to be the outward characteristic. And so when we see two letters together like this, that means that we're looking at the genotype of one individual. Remember, I said that we're working with pairs of genes here, one from the mom, one from the dad. And so that's why we have two different, what we call alleles per trait that we're looking for. So in this first individual here, they're big B, little b, and so that would be their genotype. When we're looking to determine if the individual is brown or tan, if there is at least one big B, then the dominant trait is what shows up. So this individual would be brown. And I would write out the entire word brown, but it's a little hard here on the computer. And this individual, the little b, would be tan. Here we go. We would only see the recessive trait if I have two recessive alleles. So what I did here was write out what the genotype of the parents are and what the phenotype of the parents are. Now, in a problem, you would be given, you would be given that information. Oh my gosh. Sorry, I wanted to erase that big long line, and I erased the whole thing. And so I would say step one in your problem is determining what the parent genotype and phenotype is. I rewrote it there. So a question might be, well, if we had a brown guinea pig and we mated it with a tan guinea pig, what would their potential offspring look like? Another term that we need to make sure we understand is if we have an uppercase and a lowercase, we can call that individual a carrier, and that just means that they have one lowercase in there, or they can be called a heterozygous individual. When we're setting up our cross, we'll put one allele or one letter on each of the outside of the box here. So I put the uppercase B on the top, and I put the other allele right next to it, still on the top, but in a different box, in a different column. I then took the two little b's and I put them vertically. So I separated the alleles of the parents. We're then going to combine alleles in the boxes. So this is looking at probabilities of those alleles coming together to create certain offspring. And so once we have that filled in, we need to start looking at what the genotypic and the phenotypic ratios are of the result. You can work in ratios or you can work in percentages. I usually give both on the exams. And so what we're looking at is, so we've got 4 boxes here. So it looks like we only have 2 different genotypes, big B, little b, and also little b. So we could say that we've got 50% big B, little b, and the other 50% is little b. So that would be our genotypic ratio. You could also say that it's a 1-to-1 ratio, 1 big B little b to 1 big B or 1 little b. That's fine too. You could also say it's half to half. Questions so far?
There's no rhyme or reason which parent goes on which side, right? That doesn't matter as long as they're listed correctly.
Yes, yep. As long as you're consistent, yeah, you're absolutely correct.
Okay.
Julius, how is your understanding so far?
It's good, ma'am. I do understand this one, the Punnett square.
Cool. And so once we do the genotypic ratios, I usually do those first because they'll help us determine then what the phenotypic ratios are. So remember we said the dominant trait was brown and the recessive trait is tan. In this case, we have 50% brown, which is our big B, little b, and 50% tan, which is our little b. But if we had a different problem and I had big B, then that would be a different genotype, but I could combine the big B genotype with the big B, little b genotype because that gives me the same phenotype. Both of those groups are then brown. So for any type of cross that we're doing, this is the general format that you'll want to go through. And I think having a system of figuring it out and doing it that same way each time is super helpful. And yes, definitely, brown is either big B, or big B, little b. Because it's the dominant trait, you only need one allele for that genotype to give you brown. So big B, or big B, little b is brown. And that's for the monohybrid, what I would call kind of classic genetics. As we get further into the material, and I think this will be the next live lecture topic, we'll look at some other examples like codominance and incomplete dominance, where Pooja, where that's not necessarily true. So it depends on the information that you are given. Any other questions? We can do a dihybrid cross if you like. We're not necessarily going to go into that level of detail for our class, so it's not required. Let me know if that's what you'd like to see, or we can do another kind of classic monohybrid example. Do you remember what the question was on mastering about the dihybrid? Okay, that makes sense. That was my mistake. Maybe my brain wasn't quite connected on that day. Let's see. Oops, that's not what I want. Oh, sorry. I changed my text to white, and that's why it was not showing up. There we go. All right, so the dihybrid cross is similar to what we did for the monohybrid, but we're looking at two traits. So continuing with the guinea pig example. So we know that the uppercase was brown and the lowercase was tan. So let's add long hair and short hair. So we'll do so I put the parents in the chat box. And as I'm getting this set up, if you want to start trying to pair them up, all right, so we're really looking at, well, what's the probability of offspring being a certain color and having long or short hair. And so, as Denise was saying, we just need to choose which side our parents are going to. And so maybe we can take this first parent and put them on this side here. What we need to determine is what the alleles are. So remember, we just had one letter per kind of side last time. This time we'll have 2 letters per side. We'll have one B and one S. So whenever we're working within a Punnett square and we're outside of the square, we'll have one of each. And so we can do that by using the FOIL method. And so what I mean by that is we can take this uppercase B and we can combine it with the uppercase S. So that's going to be our first one here. We also have the big B that needs to pair with the big S. And then if we, oops. And then we have the little b that needs to pair with the big S. And we have that twice because we have another big S. So that's how we set up the first parent. Our next parent, the we'll set that up on the horizontal part here. And if you've got two of the same alleles, you actually can do a shorthand. If you want to write all of it out, that's totally fine. But the only allele that we're going to end up with is the uppercase B, lowercase s. So you can shorten all of the rest of it. I could have done the same thing over here on the left-hand side too, but I wrote it all out. Either way is perfectly fine. So then when we're coming into the middle, we're putting the similar letters together. And you'll want to be consistent with your format. So I had the B's were first in the parents, and so I put those first in the offspring, and the S's were second. If there's at least one uppercase letter, then I'm going to put that first as well. So based on what we're seeing here, why don't you try to put the phenotype in the offspring? So we've got Actually, it looks like we just have two different genotypes here and here. So why don't you try to write what you think the phenotype of those individuals are using the key up at the top. And when you are done with that, if you'll give me a thumbs up in the chat box or just verbally say done. So I'll give you a few minutes to do that. All right, so I put the genotype. So we have 50% big B, big S, little s, and then 50% heterozygote individual. And so that just means I have one of each letter. So now when we're taking these phenotype, Denise, did you confirm what Pooja was saying, that they would all be brown with short hair, or did you get something different?
I got what she had.
Cool. Excellent. So they were all brown with short hair. So this was a great example of the genotypic ratio being different from the phenotypic ratio because we had those two different phenotypes, the big B, and the big B, little b, both giving us brown hair. Nice job. Julius, how did you do with that one? Did you get the same thing that they did or something different?
If you say brown, that means all the boxes contains the big B?
At least one big B. So it can either be the big B or the big B, little b.
Yeah, I got all the boxes. I got a big B, and all the boxes I got the small s. Is that?
So when you look at my Punnett square set up on the screen, did you follow that model?
Yes, ma'am.
Okay, and so we were just trying to decode the letters in terms of phenotype. So brown versus tan, short versus long hair. If you want to bring it with you to BioWork Lab later tonight, we can take a look at it as well.
Okay, yes, ma'am. Thank you.
Cool. All right. Other questions about kind of traditional Mendelian genetics, how to set them up, how to decode them?